Okay, happy Thursday evening. I've given it a little bit of time. I had a student emailing or messaging back and forth. We were trying to get the student to join, but it seems like they were unable to do so at this time. I realized that there's sometimes a lag between things that happen with the messaging in an electronic format. So we're just going to make a start now, and if the student joins us a little later, we'll reassess where we're going to be and what we're going to cover at that time. So what I was hoping to do today, earlier in the morning session, I spent a fair bit of time looking at the labs. So we had two labs for this week as it comes to the heart. So first of all, we had a look of the physiology aspect in the EKG lab, and then we had a look of the actual anatomy by dissecting a pig heart, I believe. So different groups in different combinations work through that dissection activity. So for anyone who might have missed either one of those, it is generally harder to make up a dissection activity. So I don't really know what we could do to kind of make up for that. But if you do still want to have some points from the lab, you can always submit for the EKG lab at least the portions that you can complete and in terms of a theoretical framework of the lab by simply working through the worksheet. So I provided a bunch of information about that in our morning session. So I encourage you to refer to the recording from Heart Chapter 10 a.m. morning session live online that I offered earlier today. So having said that, I figured that for this evening session, we might take a little bit of time and use it as a chance to kind of revisit the lecture content that we had for this week. So this will not cover everything and every single point that you might potentially need, but I think it will be a good start of just kind of highlighting what are some of the most important points that you might want to pay attention when you're working through the chapter material. And of course, the quiz is going to be a great way for you to then use the textbook, the interactive electronic textbook version that you've got available to work through that material as well. So I'm just going to jump through all of these and dive right in to where we started in the class. So in the class, we discussed a little bit about the fact that when you first started the study of AMP, and when you first did the introductory biology classes, I believe Bio 181 or Bio 156, you probably talked about the 4 different kinds of tissues that we had. So we had our epithelial tissue, which formed mainly surfaces and outer boundaries, covering structures, that sort of things. Then we had our connective tissues, which was a large group of different kinds of tissues, from tendons, ligaments, bone, even to as varied as blood, as we saw, as blood being a special type of connective tissue. The third tissue type that I assume that you have seen has been the muscle tissue, and we will look at the different muscle tissue types. And the final one was, of course, then the nervous tissue, nerve cells, neurons. So when it comes to the muscle tissues, what you might recall is that we had 3 different types or subgroups for that. The skeletal muscle is something that I assume that you have discussed in quite a bit of extent when you completed your BIO201, and during that course you would have covered the major muscles that generate the movement that are really visible skeletal muscles that you typically see when you study the gross anatomy of the human body. In terms of the smooth muscle, there's going to be 2 times when we're really going to dive into that. So unlike skeletal muscle that was under your voluntary control, smooth muscle is going to be involuntary. And we're going to start to see examples of smooth muscle next week when we discuss about blood vessels and how smooth muscle forms that layer around blood vessels, and that helps us to control the diameter of that vessel. So really regulate where the blood goes to what parts and where blood is needed. So all that good stuff. The third type of muscle, which as well as smooth muscle is gonna be involuntary, is the cardiac muscle. And we're gonna see the cardiac muscle only in the heart. So you don't need to think about, oh, I'd like to have a heartbeat now, or wake up in the middle of the night every now and then, and oh, I need a heartbeat. So that's why it's involuntary. But when we look at the cardiac muscle, we're going to notice certain distinct characteristics, even on a cellular level, that can be explained by the functions that we have for the heart and the muscle tissue that makes contributes to the heart muscle wall. One of the important side notes, do remember the cardiac muscle is only found within the heart. So one thing that's different from what we're seeing elsewhere with these 2 other types of muscle tissues is that cardiac muscle cells tend to be branched. I think my screen is a little overly sensitive, so let me just fix this line here so we get a little bit of a better diagram. So unlike many other cells, cardiac muscle cell has multiple branches. In addition to having multiple branches, at the end of these branches, we find a structure called intercalated disc. Intercalated discs and these branches actually serve an important purpose that can be understood by thinking, what does heart tissue do? Well, one important thing for the heart tissue and heart as a whole is that the structures that are closed by those tissues, they should be contracting at the same time. There would be no point of having a heart and having tissues, muscle tissues that help to contract the walls of the heart if each cell would be doing its own thing and contracting whenever it wishes, regardless of what others cardiac muscle cells would be doing. So these branches help a single heart muscle cell to connect to multiple other ones, and these intercalated discs actually facilitate for the materials and signals to move from one cell to another much more easily than they would typically move if they would have to cross that plasma membrane, that cell membrane. So that's the 2 adaptations that we see that help the cardiac muscle tissue to contract in a uniform manner. A couple of other things that we see with the cardiac muscle cells. One, typically central nucleus, and we still keep seeing those striations that run through the heart muscle cells. And I believe that you've talked about those striations when you looked at the skeletal muscle. So I'm going to just leave it to that. If you have any unclarity about that, you can always message me. And I do have quite a few videos available that might also guide us to that. So I'll try to make a note of that for myself. To link at the end of this video, a video that discusses skeletal muscle and striations. So, having said all that, there's one other important characteristic that I think is very striking for the cardiac muscle cells that I wanted to discuss, and that's going to be the great number of mitochondria that we find present at the cardiac muscle cells. Well, if you wonder why would we see a lot of mitochondria, let's go back to the basics and remind ourselves of the role of the mitochondria in a cell. For the longest of time, we did teach that the mitochondria was the cellular powerhouse responsible for making ATP, and that is true. The truth is, though, that we are now finding out that ATP is made also in other parts of the cell, not in as great quantities, so mitochondria still remains as the key important structure, but life isn't as black and white and as single dimensional as textbooks have previously said, so we might need to come back and update some of those. But these mitochondria serve an important purpose. They serve a purpose of making ATP within the cell, and in this case, within the cardiac muscle cell. Well, this can be explained by remembering that cardiac muscle cell is always at work. So it's going to be using a great amount of resources, great amount of energy. So for that reason, we end up needing quite a few mitochondria, more than normal in a normal average cell, present in the cardiac muscle cell. You can think of it, heartbeat starts early on after fertilization, and really when it ends, heart stops beating at the end of life. So heart really never gets to rest. So that's why we're making sure that we have this ability to supply a great quantity of ATP. I think that that's a kind of a good introduction to what I wanted to say, and we're ready to jump on to looking at the heart in its location. So heart is located between the second and fifth rib. So if you need to get an access to the heart between the second and fifth intercostal space, might be where you want to be looking at. And really, when you're trying to locate the heart in the patient's test, early on, if you're uncertain, just count the number of the ribs, and that will guide you quite far. Another thing that we see with the heart is that typically students go and look very far to the left within the thoracic cavity, but heart is fairly close to the mid-sagittal line, just slightly left of it. Locating the heart and discussing that gives us a chance to practice some of the good anatomical terminology. So we remember that heart would be located superior to the diaphragm that I have mapped on this diagram with this bluish light. So above the diaphragm, that muscle sheet that separated thoracic and abdominal cavity. Posterior to the sternum, which we saw there as a sign of or good signal of the mid-sagittal line of the body, where that would be located. Of course, anterior to the vertebral column. So heart is located anteriorly in relation to that, and just slightly left of the middle. Heart is surrounded by lungs, and we'll come back to that and the effects of that in just a few moments' time. But what I wanted to do first was to discuss about the size of the heart and how heart is positioned within the human body. So sometimes I get asked, what size heart does my baby have, or this and that person of this and that age? It is a little bit of an overgeneralization, but one rule of thumb that you can use is that if you compare the fist of an individual, that gives you a good clue about the size of their heart. And since we're looking at the fist, the other thing that I wanted to discuss was the fact that heart is really not aligned within your body, within any of these traditional planes that we have looked at. So instead, heart is in kind of an awkward angle, and let's have a look of that a little bit. So one thing that you'll notice here on this diagram on the right-hand side of the screen is that if you have a little bit of a fade of an eye, you'll notice that the heart is almost like a cone-shaped structure. You sometimes need to have a little bit of creativity when you're imagining these anatomical structures, but I think we can see rough cone-shaped structure there if we imagine. And that's especially with the role of the ventricles. So this cone has a line that runs through it from the middle. And what we'll notice is that that line is not going to be aligning to what we're used to. Well, if we take back our fist and point out our pinky finger, you can consider the pinky finger as an extension of this line that we're seeing here. So kind of pointing out from the heart. In fact, this sharp edge that we see here of the cone has the sharp edge, that would be called our apex of the heart. Many different organs have apex. Apex is the kind of a pointy, sharp bit. So we're going to imagine that in our model of the heart, the fist, the pinky finger that's pointing out is where this line coming out of, shooting out of the apex would be. And we're going to position the heart, imagine how it would be positioned within your thoracic cavity. So it is fairly close to the midline, just slightly left from it. The apex of the heart is pointing to the left, to downwards, so inferiorly, and to the anteriorly. So it's pointed in this kind of an awkward angle. And that really shows why when we discuss about the heart, when we're studying the structure and orientation of the heart, we often end up considering heart independent from the rest of the body. So we just take the heart away from those traditional planes and we consider it separate from the rest. So I hope that that makes sense. Another thing that this diagram shows nicely to us that we will come back to is the two ventricles of the heart. You will see the left ventricle with its thicker muscle wall, whereas the right ventricle has much thinner muscle wall here. So this will become important as we discuss about the role of these ventricles just a little bit later. The other thing that I wanted to take just a moment to discuss at this point is the fact that heart is located inside a sac, what we call pericardial sac or pericardium. This sac serves a few different purposes. First of all, if there's an inflammation somewhere in the body, it tries to these compartments of the body to separate that the inflammation cannot spread wildly throughout all parts of the body. And that's true also for the heart. So we don't want heart to get inflamed. We try to separate it from the other parts. Also, you can think of this sac as giving us a little bit of space. The heart is there stuck between the lungs. So, and heart needs to move. So we kind of try to ensure that we have a little bit of room for that by having the heart inside this pericardial sac. So let's have a look of the pericardium and what we can tell about this sac. Well, pericardial sac is made of actually 3 different structures that I want to pay attention. First of all, there's going to be 2 layers. The more external layer is going to be our parietal layer, and the more internal layer is going to be our visceral layer. And between those two, there's a pericardial cavity, this space. Within this cavity, we do end up finding a little bit of fluid, and that fluid serves the purpose that it allows these layers to slide past each other smoothly without friction. So that's our pericardial pericardial sac. And one thing that you can sometimes end up noticing or hearing is if there is an inflammation within the pericardium, you might actually hear that. simply by listening with a stethoscope. So this condition of having an inflammation, so remember the term itis in medical terminology referred to inflammation of something, and in this case pericarditis, inflammation of the pericardium. Well, that can be heard with a stethoscope as this kind of a creaking sound when you listen with a stethoscope, that not everything's sliding nicely how it should be. So that's our pericardium. There was in the previous picture something else that I wanted us to spend a little bit of time as well. So let's have a look of that. So what we see here is the heart wall, the actual muscle wall of the heart. And we'll notice from this diagram that it's actually typically divided into 3 layers. And epicardium forms this outermost layer of the heart muscle wall, and endocardium the innermost layer of the heart muscle wall. And I sometimes try to think of how can we remember these terms. Can I offer any tools for our students? And I'm going to give it a shot. Let's see if it helps. So we're going to start with the epicardium. So I always try to connect the terms that I'm introduced with something that I would know from the past. And one thing that I've done in the past has been that I've studied quite a bit of geography and geology. And one thing that we see when we look at the Earth is that earthquakes really rarely happen right at the surface of the Earth. They happen a little bit further deep within the crust of the Earth. So when we're describing where the earthquake took place, we will map up from that to the surface of the Earth, and that spot directly above where the earthquake actually happened is called our epicenter. So that gives us a clue that epi refers to something on the surface. So similarly, epicardium is going to be the layer at the most surface layer of the heart's muscle wall. I hope that that helps. Endocardium instead is the innermost layer. You can think of it endo, it's the end of it, it's the innermost, it's endo, the inner layer, whatever helps you to remember that. And in between these two is the thickest and the fleshiest part of the heart muscle, which is known as myocardium. And this myocardium is really what I've been super interested in my research and on the work that we've been working on with the research group on that. So I think that those were the most important things that I wanted to highlight from this particular picture. A good picture, quite a bit of information going on there. We already had a look of the pericardium. Well, what we're going to do next, we're going to cover quite a bit of concepts. We're going to look at the heart. We're going to look at a simplified diagram and make it a little bit anatomically more correct. That will help us to separate or learn different parts of the heart. And we will also discuss the flow of the blood through the heart. But before we get into that, I wanted to remind us of the basic concept that sometimes human heart is described as being 2 hearts beating as one. So what you'll notice is that we have left and right side of the heart. And remember, this is going to be now left and right as it relates from the viewpoint of the patient. So we're going to have here the right side of the heart, and here the left side of the heart. And what you'll notice is that the right side of the heart deals with deoxygenated blood. We discussed about that in our last chapter where we looked at the blood. So this blood is low in oxygen concentration. So it deals with the low oxygen concentration blood. Whereas the left side of the heart deals with blood that is rich in oxygen, high concentration of oxygen on the left-hand side. And both of these pump in a similar manner at the similar rhythm. So now that we know that there's going to be 2 sides, that gives us a clue that we're actually going to be looking at 2 different loops or 2 different circuits where the blood is going to travel in the body if we look in general level. Kind of simplifying So let's start with the first one, which is going to be our pulmonary circuit. So blood from the right-hand side of the heart will travel to the lungs, get reoxygenated there, and return back to the heart as rich in oxygen. This loop going from the right side of the heart to the lungs and returning back to the left side of the heart is known as pulmonary circuit or pulmonary circulation, referring to the lungs. The second circuit that we can see then supplies all other parts of the body. So that's going to supply blood that's rich in oxygen from the left-hand side of the heart to the body, where oxygen is picked up by tissues and the blood returns low in oxygen back to the heart. It's actually going to be carrying carbon dioxide in great quantities, and then that goes back to the loop, to the pulmonary loop, and gets offloaded at the lungs. But what we see is that the systemic circuit then supplies all other parts of the body than the pulmonary circuit. Well, now we need to think about where do we find more blood vessels, what are the size differences, what are differences between these circuits. And one thing that will stand out immediately is that lungs are very close to the heart. So there's not a long distance for us to go to get here. So for that reason, the right side of the heart doesn't really need to contract super hard to pump the blood to the lungs. The distance is short, and it's not really a great force that we need there. However, there's going to be a lot blood vessels where we are pumping the blood from the left-hand side of the heart to the systemic circuit. So to make sure that blood goes to all of these parts within the systemic circuit, that's going to take more force. So we end up seeing that the pressure with which the blood is pumped out of these two sides of the heart is going to differ. So on the right-hand side to the pulmonary circuit, the force is going to be lower, so we have low pressure there, whereas on the left-hand side pumping to the systemic circuit loop is going to have very high pressure on the blood. So I hope that that makes sense. And that kind of prepares us to start looking at the heart in a greater detail. And here's a really nice anatomically fairly accurate drawing of the heart. We have removed half of it and we're kind of peeking in. And what we can see is that it should be fairly obvious here that there's going to be 4 chambers, one here, one here, one here, and one here. So we're going to have 2 chambers at the more superior part, top part of the heart, and 2 at the bottom part, so more inferior part of the heart. You can also notice how there's a difference in the thickness of the wall on these lower chambers. And you'll notice that on the left-hand side, this wall is much thicker. So rather than drawing this very fancy, sophisticated diagram, you can simplify your life just by all means, if that is what rocks your boat, and draw a heart as such. As simple a box with 4 different sections. Each one of these represents of each of these chambers that we just saw here. So here, 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 and here. So it's a little bit of a simplified way of showing what happens with the heart. And let's use that to review the different parts of the heart. So the top parts, these 2 top chambers, we're going to refer to them as atrium. And atrium, I think that one way to remember the name is to compare it to the atria of the buildings, or atriums of the building. These are kind of the receiving spaces between when you come into the building from the outside before you enter to the offices, kind of like entrance halls, if you wish. And ventricles are then going to be the more inferior chambers. They're the ones that actually don't just collect blood, but work really hard to pump the blood out of the heart, whether it is to the pulmonary or systemic circuit. And then we named these based on the side where we're at. So we're going to have right atria and right ventricle, and left atria and left ventricle. So blood arrives to the heart, to the right atria from systemic circulation. So this blood is now going to be deoxygenated, and it arrives through superior vena cava, which drains the head, neck, and upper limb, and through inferior vena cava, which drains the remaining parts of the body. Blood collects to the right atrium, and then once the right atrium is ready to contract, gets passed through our right AV valve to the right ventricle. Another name that you see sometimes being used for the right AV valve is tricuspid valve. Well, then when the blood has moved from the right atrium to the right ventricle, this valve is going to close so that the blood cannot return back to the direction where it came from. So we're establishing this unilateral flow of blood. That's going to be important for us. So blood only travels within the heart and within the human body to one direction and one direction only. Well, now we have all this blood at the right ventricle. Remember, it's still deoxygenated, low in oxygen, and we want to pump it to the lungs. How we do this is that the right ventricle starts to contract. Blood cannot return back to the right atrium, so it has to shoot out to the pulmonary trunk, which opens from the right ventricle. Pulmonary trunk would run in front of the right atrium, so the blood doesn't go back to the right atrium. This is not probably the best diagram I've ever drawn. You can think of this line here, maybe it's a dotted line. So it's just shooting out from the right ventricle to the pulmonary trunk. There is going to be a valve there separating the pulmonary trunk and the right ventricle to ensure that blood cannot return back to the right ventricle from the pulmonary trunk, from the lungs, to the direction where it shouldn't be coming. Well, now the blood has gone to the lungs, it's gotten reoxygenated there, and it returns high in oxygen to the left atrium. So left side of the heart, there's actually 4 openings for the blood to come in there, and once blood has pooled there, kind of build up there, it travels from left atrium to left ventricle through another valve. This is going to be our left AV valve, or sometimes referred as a bicuspid valve. Another term that you might see being used for this valve might be mitral valve. And again, its role is to prevent that the blood cannot flow back from left ventricle to the left atrium. So now that we have emptied the left atrium, which was full of blood that's rich in oxygen, it has traveled to the left ventricle, and left ventricle starts to contract. Blood cannot return back, so it has to shoot out from the left ventricle, and it shoots out to aorta. Again, the blood doesn't go back to the left atrium or through it, so aorta is separated from left ventricle with an aortic valve. And now from the left ventricle, the blood goes to the systemic circulation, supplies the body, all tissues within the body with oxygen until it's deoxygenated and returns back to the right atrium. So that's the short loop that we just had a look of, and the chambers that we have now added to this discussion. So let's have a look of that with a little bit more anatomically correct representation of the heart. So we see the 2 atria. So we see the right atrium and left atrium here. And then we can see our ventricles, right ventricle and left ventricle. So remember, blood coming in through superior vena cava and inferior vena cava building up to the right atrium. Then from there, through the right AV valve, or sometimes known as a tricuspid valve, to the right ventricle. When right ventricle contracts, this valve closes. The blood cannot return back to the right atrium. Instead, it has to go up to the pulmonary trunk. And it's going to go through the pulmonary valve. And again, the role of the pulmonary valve is to ensure that blood cannot return back to a direction that it shouldn't be flowing. And from the pulmonary trunk, this deoxygenated blood, notice it's all deoxygenated on the right hand side, goes to the lungs, gets reoxygenated, and now it returns from the lungs high in oxygen to the left atrium. From left atrium, blood travels to the left ventricle as the left atrium contracts through our left AV valve, or bicuspid valve, or mitral valve. Many names there that we can use. And once the left ventricle starts squeezing, this left AV valve closes, and blood has only one way to go, which is going to go to the aorta, and it's going to go through aortic valve. And aorta carries the oxygen-rich blood to all parts of the body until the oxygen has been picked up, and it returns back to the right atrium as deoxygenated blood. So that was a quick revision of what we just learned with a little bit more anatomically correct diagram. A couple of parts that I do like asking from my students is the wall that separates the two ventricles, and the clue is in the name, interventricular septum. So septum in anatomy, and anatomy especially, refers to a wall of some kind. So it's going to be a wall, and it's going to be interventricular septum. So it's going to be a wall between the ventricles. So here, that's the explanation for the name interventricular septum. I like asking that from my students, so you might want to make a note of that, and also make a note of that apex that we discussed earlier on, the peak or the sharp edge of the heart. So the valves, let's have a quick discussion, just revisit those from what we have already discussed. So we had AV valves. AV valves separated the atria and ventricle on both sides, on the right-hand side and the left-hand side. The other names that we might sometimes see being used actually come from the shape of these valves. So what we have here at the left upper corner of the screen is a diagram or a drawing that shows the heart as if we were looking down on it from the top. And we can see four important vessels. We're going to be focusing initially on these AV valves. So here I have circled the right AV valve, which would be here, how you can see it from the top of the heart. You can actually see that it has three leaflets on it. So three cusps. That's where the term tricuspid valve comes to describe this valve. Similarly, on the left-hand side, the left AV valve, it actually is made of two cusps, two leaflets, so bicuspid valve. The other name that I mentioned that sometimes gets used is the mitral valve. And I've been always wondering where does the name mitral come from? And actually one of the students in your class went through the trouble of finding out for me very kindly. So apparently the term mitral comes from a Latin word referring to the hat that the priests have. So it's kind of disc-shaped headgear. So I guess that mitral valve, some can see some resemblance there to this headgear that the priests would be wearing. So I don't know if you can see it there. I'm not totally sure, but I certainly appreciate knowing an explanation where the name is coming now, at least. So that might be a fun curiosity fact to know. So that was our AV valves. Let's have a look of the pulmonary and aortic. So pulmonary valve separated our right ventricle from the pulmonary trunk, and aortic valve separated our aorta from our left ventricle. So simply named after the structures that follow them, pulmonary trunk and aorta. Sometimes these two valves are both described as being semilunar. And I think that the term semilunar again refers to the shape of these valves that we can see here. So it would be referring to these two. Semi referring to partial, and lunar referring to moon. So its shape looks in someone's eyes as partial moons. I'll let you choose if you can see that there or if you cannot. But that's the short story behind these names. So those were our valves. You might ask me sometimes, or students ask me sometimes, do I need to know all of those? And unfortunately, all versions would be what I would expect you to know for each of these valves. So what else? What else can we see? We can see the diagram that we just had a look of in terms of the simplified version. And I wanted to discuss about these valves a little bit. So not always valves work perfectly. The main idea, main role of the valves was, of course, to facilitate unidirectional flow of the blood through the heart. So we want to establish one-way flow only. So we have these valves that have the leaflets. And in case of the AV valves, there's a lot of force from the ventricle. So these valves are actually going to have a special adaptation. And this is only applicable to the AV valves. We have papillary muscle coming out from the wall of the ventricle, and then little strings called chordae tendineae that connect from the papillary muscle to these valves. This is to prevent that if the force is really strong and the blood flowed only this way, if the force is really strong and pushing backwards to these valves, that force will not break the valves. They won't flip backwards and fail in their function. So that's what happens normally. We don't see these adaptations, papillary muscles and chordae tendineae in the semilunar valve, so pulmonary and aortic valve. But another thing that we might want to pay attention is that sometimes the valves don't work correctly. They might be limited in how much or how well they close. So being always a little bit open, in this case, in incompetent valves, some of the blood actually gets to sneak backwards as well. So we're pumping blood back and forth a little bit. That would be a problem. We're not efficiently moving the blood through the one direction as we should. Valvular stenosis instead refers when these valves don't open properly, so they're a little too stiff. So heart has to work extra hard to squeeze the blood through them to the right direction. So that's less than ideal as well. We can try to fix valves that don't work either by getting a valve from another individual, a dead catheter, and implanting that, or getting a valve from another animal. Pig heart valves are very commonly used, or by using a mechanical heart valve. The mechanical valves are not quite as common. There's a bunch of problems that comes with, just like hinges of the door. Think of the valves, how much they have to move throughout the entire lifetime. Everything wears down over time if it's a man-made. So there can be failures on those. The other thing is that the mechanical heart valves are not able to respond to the changes in the size of the heart. So for example, we can't put them for very young patients because they wouldn't be able to keep up with the change in the growth of the heart. What I want to do as the last thing when it comes to the anatomy of the heart is to discuss just a little bit about the surface anatomy of the heart. And there's few things that I want to highlight. We're going to start with the coronary sulcus. That's often known also as atrioventricular groove. And that's this groove, this depression. You might remember the term sulcus from the surface anatomy of the brain. So depression of some kind. This is the depression that runs between atrias and ventricles. So this nice depression here. And the reason why we're studying these surface anatomy features is not so much these features, but what will be running in them. So just hold on to that thought and we'll come back to that in a little bit. The other depression that we can see is running here on the anterior side of the heart between the ventricles. So it's called anterior interventricular sulcus. So the depression between the two ventricles on the anterior side. And we have the same thing on the posterior side as well. I just haven't included a picture of that, but similar depression between the ventricles. So we have posterior interventricular sulcus as well. So the reason why I wanted to talk about this is that they're going to be important when we talk about coronary arteries. So just like any other tissue in the body, heart muscle cells require constant supply of oxygen and nutrients. Remember, they were working really, really hard. And what we have here actually is a really nice picture of all the blood vessels on the surface of the heart itself. And there's a lot, like you can see. So you can imagine that heart muscle cells would be really hungry for oxygen and nutrients. They're working real hard there to do their job. So there has to be a good blood supply. And this blood supply is actually through these two little blood vessels that appear immediately after the aortic valve. And these are going to be our coronary arteries. There's going to be one on the right-hand side and one on the left-hand side. So let's have a look at those. So first of all, the left coronary artery appears on the left-hand side of this part right after the aortic valve, and it divides real soon into two branches. The first branch that we see runs in this anterior interventricular sulcus that we had a look of, and it's going to be called anterior interventricular artery. So it runs to the apex of the heart, supplying the anterior portion of the heart muscle wall and the apex. Sometimes we use in medical language another term for this, which is left anterior descending artery. So you might see it being abbreviated also as LAD. The second branch of the left coronary artery is actually going in this groove between the atrium ventricle on the left-hand side and loops around the heart, encircling it to the posterior side. You can see it, it's kind of drawn on a paler color as if seen through the heart. So it goes to the behind the heart. Since it loops around and circles the heart, we call it circumflex artery or left circumflex artery. And it runs on the posterior side for the posterior interventricular sulcus. The third blood vessel, the coronary artery that I wanted to discuss, is going to be our right coronary artery. It arises on the right-hand side and runs in this atrioventricular groove on the right-hand side and loops again around the heart to the posterior side, and then runs and meet then at the posterior interventricular sulcus. Which one of these runs down on the posterior interventricular sulcus varies. That's individual variation. So that's where we start to see that not everyone is as equally designed or similar. So some are more dominant for one of these arteries that run on the back of the heart. So those were our coronary arteries. There is going to be also coronary veins that drain the blood away from the heart muscle. I'm going to leave that for you to study independently. And that concludes everything that I wanted to say in terms of the anatomy of the heart. So the only thing we are left to discuss about is going to be the physiology of the heart. And there is quite a lot to discuss, and I'm going to try to keep it brief in the interest of time, but let's have a look at that together. And before we get too far, I want to remind ourselves from a concept that you learned when you studied the nervous system on BIO201. So you learned about the action potential in the nerve cell. So one of the things that you probably learned was that there was a resting potential that was negative 70 millivolts inside the nerve cells. So you remember this pumping of materials across the cell membrane in the nerve cells, and this pumping resulted in an imbalance, which meant that the inside of the nerve cell was about 70 millivolts negative. That's just the normal state of being of a nerve cell, and that was called a resting potential. Well, if there was a need to pass an electrical message within the nerve cell, what we saw was a change in this charge temporarily at the place where the message is traveling. So it travels through the axon or through the nervous system, and this change will be seen. So the change that takes place is that the nerve cell actually depolarizes. So it goes towards zero and a little bit above it even. when the message is passing, electrical impulse is passing through that particular spot. There is a little bit of a dip, but then a plateau stage where the charge remains either at the zero or close to the zero before it returns back to the negative resting potential, so it repolarizes. One thing that you might remember during repolarization, we had this part where the charge actually for a brief amount of time went even below -70. This undershoot was there to ensure that another action potential couldn't pass immediately after. So we had kind of this recovery time, refractory period. And then the action potential was gone and the resting potential of the nerve cell returned to -70. So that was in a typical nerve cell that we had a look of. And one thing that you'll notice in the nerve cells, these action potentials take only a very briefly amount of time, so between 2 and 5 milliseconds or so. Well, let's have a look of what we see in the heart then for the cardiac action potential. It has some similar appearance, but it's almost like it's a little bit tilted. So the resting potential instead is constantly climbing towards a threshold of -40 millivolts. And once it reaches the -40 millivolts, an action potential is triggered. So this explains why the heart never stops beating. It has this constant trigger because it's constantly climbing even in a resting state towards that trigger spot. Then action potential takes place and we go to the recover and next action potential takes place. Probably the most important thing, other than looking at the shape that you might take away from this, is when you look at the duration of cardiac action potential. You'll notice it's much, much, much longer than the nerve cell action potential. So cardiac action potential somewhere around 200 to 400 milliseconds. So significant difference. And this delay is important so that the blood has time to leave one chamber and travel to the next. We don't want to rush the blood. If the blood doesn't move from one chamber to the next, we're really not doing anything useful as a heart. So let's have a look of how these cardiac action potentials or these action potentials within the heart travel. And for that, we have included here a little bit of a picture of the heart cut in half, showing the 4 chambers, the atria, and the ventricles. And we're seeing our right side and left side. So you'll notice right at the right atria, superior portion of that, there's a clump of nerve cells and nerve cell bodies. This part is known as sinoatrial node. That's the pacemaker of the heart, what triggers the heartbeat. From there, an electrical signal travels across the atria on both right and left hand side, and as this electrical impulse passes through, that triggers the atria to contract. Now that the atria has contracted, the blood will flow from atria on both sides to the ventricles, and the action potential actually takes a little break. There's a little delay station known as AV node. It's between atria and ventricle on the right hand side. So we hold the action potential there for a moment. The goal is to ensure that the blood has left the atria and we have moved the blood from where it needs to go to where it needs to go. Well, after this little delay, the electrical impulse will travel down the bundle branch towards the interventricular septum, where it travels down to Purkinje fibers, and eventually, and once it's reached the apex, up on the walls of the ventricles. Now that the action potential is on the ventricular walls start to contract and push the blood out either to the pulmonary trunk or aorta, respectively. So really what we're seeing is this kind of synchronized atria contracts, ventricle contracts, atria contracts, ventricle contracts. So there needs to be that little delay so we have time for the blood to move from one part to another. So that's really the important role of the AV node. Sometimes these contractions don't work the way we would want them to do, and they're uncontrolled. And when these contractions are uncontrolled and all over the space, blood is not moving where it should be within the heart. Well, luckily, we have a machine known as Automated External Defibrillator, AED for abbreviation, that can be used to basically deliver an electrical shock. We put two leads on different sides of the body so that the heart is bang on in the middle of them and deliver an electrical shock in hopes that that shock reestablishes this rhythmic contraction that we were just looking earlier on within the heart muscle walls. So the goal is really to shock us back to where we should be. This can be only done if there is some kind of a rhythm or some kind of a shockable rhythm left within the heart. So if the heart is completely dead, you can just bounce it back to life with an electrical impulse. There's nothing to shock. So really the AEDs are the machines that save lives, and they are really, really easy to use. They will actually literally talk you through of everything that you need to do as you're using them. Well, some individuals have this issue happening so frequently that we can't really have them walking around with an AED. So instead, what you find these individuals having is a pacemaker, similar machine as the AED, but this is now implanted inside their body. And it has a computer part that monitors and, if necessary, delivers the shock, and metal wires that wrap around the heart so that the shock gets delivered to the right places at the right time. So that might be something that you see on some individuals. If you get a chance, it's worth of having a peek. You can actually see it through or see it as a bump externally. So having said all that, the electrical impulses are going to be important. And one of the tools to study these electrical impulses that we might use is to do electrocardiogram with a method known as electrocardiography. What we're doing here, we're measuring all of these electrical currents within the heart and kind of showing a summation of them. Typically, we would use 12 LEDs, so you actually see a better three-dimensional view what's happening in different parts of the heart, and that's why you will have multiple traces. And by analyzing this little wiggly line that shows the electrical currents that are taking place within the heart, we can look at, is everything working how it should be? Does everything look the way it should? Is it phased in the right timings? Or if there's a conduction problem with the electrical impulses, where in the heart does that take place? So we can get a great amount of information already early on from this graphical recording of the electrical activity of the heart. And here I've taken a section of a typical ECG or EKG and made it a little larger and highlighted the most vital elements of our EKG. And you'll notice that there's three parts that are especially striking for a single heartbeat. So let's go through those. The first one is going to be our P wave. The P wave tells us that now sinoatrial node and the atria are actually depolarizing. Following that, you will see a set of three spikes. This is known as our QRS complex, so Q, R, and S spikes. During the QRS complex, the ventricles now become depolarized and atria repolarizes. And finally, this is followed by T wave, which tells us that now our ventricles have repolarized. So we're really looking here, are we seeing all of the elements we're expecting to see? Are they showing up the right way, right shape? Are they timed properly how they should be? And we can study all sorts of information from these elements. And that's what your lab was all about. So I hope that you found it useful. Couple of other terminological things. Like I said, we can study distances between these elements, but a couple of other terminological things that I do want to highlight and I want you to be comfortable with because they're going to be very important when we start talking about the blood vessels and blood pressure. The period when the heart is contracting, and especially the ventricles, so blood is now leaving the heart. When it's contracted, we talk about heart being in systole. And when the ventricles are relaxed, so heart is not actively pushing blood out, we talk about the heart being in diastole. So systolic and diastolic blood pressure is what we measure when we want to measure that what's the blood pressure when the heart is contracted. So pushing the blood out, what's the blood pressure when heart is relaxed. So both of those will become very, very important. Little bit about the heart sounds. When you listen hard with a stethoscope, you will end up hearing two different sounds. Typically, it sounds a little bit like lup up, lup up, lup up. So these two sounds follow each other. And what you're actually hearing is first the sound made by the AV valves as they close, followed by the sound made by the semilunar valves as they close. So that's what we're listening with a stethoscope. And actually, if you position your stethoscope smartly to the right place, you can actually focus on each of the specific valves to hear, is that valve functioning the way we expect it to function? So we can, by listening alone, get a lot of information about what's happening and is everything working the way we want it to be working. If we hear abnormal heart sounds, we talk about hearing heart murmurs with the stethoscope, and that can tell us that we have issues with the valves, whether it's the incompetent or stenotic valve. We had a look of that before, so I'll just leave it for you to study independently. And the final thing that I wanted to talk in terms of the physiology a little bit is looking at the amount of blood that leaves the heart at one time. So the volume of blood that leaves the heart in a minute is known as cardiac output. And cardiac output is really a result of two factors that we can be looking. First of all, we can be looking at the heart rate. So how many times per minute is the heart contracting? And second thing that we can be looking at is going to be how much blood gets pumped out of the heart with each contraction. So what's the stroke volume? And these together then define the cardiac output. Typically in a resting state, the cardiac output would be somewhere around 5.25, 5.25 liters per minute. That's about 1.5 gallons. I apologize, 1.5 gallons. If you wonder how amazing organ heart is, I think it helps when you realize that the cardiac output can go up from 5.25 liters per minute to even to 20 to 25 liters per minute if there's a high need. And this is done by increasing the output in such a significant manner. So that completes the lecture material that I had planned for this session of ours to kind of review in a very quick, very brief. I hope that you found it useful. Do remember that you have full lectures available also as recordings on your Canvas course. So those might come in handy. I can't think of anything else to add or share other than just to remember to do your homework. So your heart quiz, heart practice anatomy, and then the lab. And we had 2 labs for this week. We had the EKG lab, which is the worksheet that you will actually find on the Canvas. And you did also the heart dissection, which I hope will help you and you will find interesting. But like I said, really for the heart dissection, you kind of needed to be there. So if you're interested about the labs, check out the video from this morning. I hope you will find that useful. And as always, I am here to help you every step of the way. So do not hesitate to reach out to me. I hope you have a very good rest of the week, and I look forward to seeing you next week.